Hello everyone. In today's video, I'll be dealing with a topic called glycolysis, which comes under the respiration. It is one of the very important topic when we deal with the subject respiration. It is also called as Emden, Meyerhoff and Parnas pathway to honor the scientist who elucidated this pathway in detailed manner in the past. Glycolysis is a term which is basically having two terminologies like glyco and lysis, and glyco refers to sugar and lysis refers to breakdown. That is, it is the a catabolic reaction which tells about the name and the breaking down of the sugar molecule for the purpose of respiration to get the energy. And glycolysis is a cytoplasmic pathway which breaks down glucose into pyruvate. This is what we are going to study in this particular video. And this breakdown happens in the sequence of 10 reactions, and these 10 reactions have been classified as investment phase. That is, the first 5 reactions are called as investment phase and next 5 reactions are called as payoff phase. And let us learn these reactions one by one. So let us focus on investment phase. Why it is called as investment phase? Because here ATP will be gets utilized. So we have to the cells will utilize the ATP instead of production. That's why it is called as investment phase. So before I begin my series of reactions, I want to make clear some of the structure of this particular reactions I have used. So first I have named the reaction, numbered the reaction. So in coming slides also you can see that here will be the number, reaction number or stage number, step number will be there, and generally called name will be written here, and product will be written here, and beneath the product you can see that number of carbon atoms, because in glycolysis, since it is a breaking down reaction, there will be less carbons will be having. That means a 6 carbon compound will become a 3 carbon compound. Along with that we have also named the phosphate groups in a rounded shade. And here the bottom line, whatever the names are there, that will be of the enzyme which is involved in that particular reaction, and these arrows will suggest us whether the reaction is forward reaction or reversible or irreversible reactions. And here the other substances whatever involved here, they will be depicted in this region. So with this basic background we will start our study of glycolysis one by one. The first reaction of the glycolysis is called as phosphorylation, because here the glucose gets phosphorylated by a phosphate group received by the ATP molecule. ATP refers to adenosine triphosphate, and this ATP molecule under the influence of the enzyme hexokinase gives rise to glucose 6-phosphate, which is a 6 carbon compound with 1 phosphate group, and this particular reaction is irreversible. And here we will get ADP and proton as a products along with the glucose 6-phosphate, and also to mediate this particular reaction Mg2 plus or magnesium will act as a cofactor. This is what we have to study about this particular reaction. Next, second reaction is called as isomerization, where the glucose 6-phosphate gets converted into fructose 6-phosphate. As we know that glucose is a aldose and fructose is a ketose, so in between them isomerization takes place with the help of enzyme phosphohexose isomerase, and here the functional group shifts from aldose to ketose. So isomerization takes place, and this will help to move the reaction forward. But this reaction, depending on the condition, can be moved backward also. So it is a irreversible reaction. Sorry, reversible reaction. And then third step, again one more phosphorylation takes place where fructose 6-phosphate, that is one 6-carbon compound with one phosphate group, will become fructose 1,6-bisphosphate in the influence of the enzyme phosphofructokinase. Here in the position number 1, one more phosphate will be added by the ATP, and which becomes ADP, and here also magnesium acts as a cofactor. Further moving, fourth reaction is generally called as cleavage reaction, where it gets split into 2 3-carbon compounds. Here fructose 1,6-bisphosphate under the influence of aldolase. The aldolase may be different in different kind of organisms like bacteria, plants, animals. So generally we call it as aldolase to keep it very simple. Here the 6-carbon compound fructose 1,6-bisphosphate will break down or split into 2 carbon compounds of 3 carbon compounds, and 1 carbon compound is 3-phosphoglyceraldehyde and are abbreviated as 3-PGAL, and dihydroxyacetone phosphate, DHAP. It is also DHAP, it is also abbreviated like that. So here it's very important reaction because the first breaking or split of the molecules takes place here. And the fifth reaction, what happens? So again isomerization happens where the dihydroxyacetone phosphate prefers to get back into 3-phosphoglyceraldehyde. That is, it's another isomer in the presence of enzyme triose phosphate isomerase. So this further all the reactions of the glycolysis will be forwarded only by 3-phosphoglyceraldehyde. So whenever 3-phosphoglyceraldehyde is produced, the reaction will continue. If that doesn't get produced or dihydroxyacetone phosphate gets produced, the reaction will halt. So this is how the glycolysis also takes its own control measures in various manners, but this is also one among them, and we are not discussing its control measures and all maybe beyond this particular topic, and let us focus on the preliminary reactions with their names and enzymes involved in with their nature. Okay, so far we have learned about the preparatory or investment phase reactions in the glycolysis. Now let us have a view on payoff phase. That means the real energy molecules being generated in the glycolysis, because ultimately the aim of the glycolysis or aim of the respiration is to produce the energy. So that's why we have to be very focused on these issues, and here let us learn that the payoff phase of the glycolysis have 6 more reactions. Here ATP and NADH gets generated. Let us study how they get generated in which reactions. 6th reaction, dehydrogenase reaction, where the 3-phosphoglyceraldehyde will become 1,3-bisphosphoglycerate. Actually this reaction is a little bit complicated reaction, but let us focus on these two, this reactant and this product, and let us minimize our understanding about this by understanding the enzyme involved in that and the overall process which is involved that for this particular discussion. So here what happens actually? So actually 3-phosphoglyceraldehyde will combine with an inorganic phosphate. As a result that the one more phosphate group will be added to this. Otherwise there is no source. Earlier in the investment phases, ATP used to give the phosphate group, but here there is no, ATP is not giving phosphate group, so inorganic phosphate is acting as a substrate or reactant to give phosphate group. And to achieve this particular reaction, there should be a dehydrogenase reaction should takes place with the help of glyceraldehyde phosphate dehydrogenase enzyme, which makes NAD to undergo reduction to form NADH. So nicotinamide adenine dinucleotide is the name, the full name of the NAD. So when it is in oxidized state, we will mark it as a positive, and when it is reduced, we will mark it as a H. So this is the oxidized form, this is a reduced form. So reduction takes place, dehydrogenase in the presence of dehydrogenase enzyme, and as a result we can see 1,3-bisphosphoglycerate will be produced. Again, there two phosphates have come into picture. This will bring out a marvelous reaction, that is the seventh reaction, and it is called as substrate level phosphorylation. So at this point of time, the glycolysis will come to the level its lost ATPs, because glycolysis already have spent 2 ATPs in the reaction number 1 and reaction number 3, but here only the 2 ATPs will be generated, because sixth reaction onwards there will be 2 components of each reactant will be there. And here 1,3-bisphosphoglycerate undergoes substrate level phosphorylation. What is the meaning of substrate level phosphorylation? Substrate level phosphorylation is nothing but a phosphate group will be react from the substrate, will participate in the reaction with the ADP or GDP, but AD here in the case ADP. As a result, the ADP gets one phosphate and it becomes ATP, which is really needed for a cell to work. And this particular reaction is also mediated by the the phosphoglycerate kinase and Mg2 plus as a cofactor. As a result, we will get 3-phosphoglycerate. So in sixth reaction, we have to recall that it was a glyceraldehyde, but it became, at the end of the sixth reaction, it becomes glyceric acid or glycerate. Now this 1,3-bisphosphoglycerate loses its one of the phosphate group and to form ATP, and it becomes 3-phosphoglycerate. Next, what happens? Next, isomerization takes place. Though the name mutase enzyme is phosphoglycerate mutase, but here what happens? Just positional change will takes place. So 3-phosphoglycerate becomes 2-phosphoglycerate. So positional shift takes place, and this is one type of isomerization only, and it leads to the little bit destability in the reaction, and as a result the reaction will move further. By the way, you might have noticed there is a symbol, sixth reaction onwards there is a 2x symbol, which again and again tells about that all these reactions will happen for calculation twice. That means 3-phosphoglycerate molecules will get converted into 2,2-phosphoglycerate molecules under the phosphoglycerate mutase and the influence of the phosphoglycerate mutase, and so on. The ninth reaction of the glycolysis is dehydratase reaction, where water gets removed and 2 Mg2 plus cofactors will work on 2-phosphoglycerate under the influence of enolase enzyme to produce phosphoenol pyruvate. So again, there is no structural change, positional change of the phosphate group here, but other structural changes will take place among these with respect to these two carbon molecules. As a result, the phosphoenol pyruvate gets ready for its final show of one more substrate level phosphorylation. That is the 10th reaction, last reaction of the glycolysis, which undergoes, where phosphoenol pyruvate undergoes substrate level phosphorylation. That means the phosphate group of this phosphoenol pyruvate will get added with the ADP to produce ATP, where the magnesium acts as a cofactor, and ultimately we get the pyruvic acid or pyruvate, which is the final product what we are expecting in the glycolysis. So this is how the 10 reactions of the glycolysis can be studied with respect to their reactants of the each stage, products of the each stage, and nature of the reaction, enzyme involved in that, and all other assimilatory powers which are involved then along with the cofactors, coenzymes, or any other water molecules or inorganic phosphates, as in the 6th reaction, like that. So this is the overview of the glycolysis what we have studied so far. Let us have a special note on the enzymes which are performing the function of glycolysis. Basically, there are 4 types of enzymes which are involved to run the show of 10 reactions. Among them, the lead role is taken by the transferases enzyme, where usually you can see the end with the term kinases. So the 1st, 3rd, 7th, 9th reaction of the glycolysis will be carried out by the transferases, where they are the class of the enzyme that enact the transfer of specific functional group from one molecule to another. So the molecule which gives the functional group, just like in the hexokinase, what happens, the phosphate group will be added. So like that, so it will be called as donor and acceptor molecules in both the cases. So another set of enzymes which are used in the glycolysis are isomerases, where they convert a molecule from one isomer to another, and the isomerases facilitate intramolecular rearrangements in which the bonds are broken and formed. So 2nd, 5th and 8th reaction of the glycolysis is carried out by different enzymes, but categorically they come under isomerases. And the 3rd class of the enzymes which will involved in the glycolysis are lyases. So there are 2 enzymes, aldolase and enolase. So these enzymes are involved in catalyzing the breaking of various chemical bonds other than hydrolysis and oxidation. So they have mechanisms other than hydrolysis and oxidation. So they are comes under lyases. Last but not the least, oxidoreductases is enzyme which helps in the 6th reaction of the glycolysis, that is the production dehydrogenation glyceraldehyde phosphate dehydrogenase, where it converts 3-phosphoglyceraldehyde to 1,3-bisphosphoglycerate. And these reactions will act as electron donors and acceptors and oxidoreductases. So oxidation and reduction also takes place here. So with this note, I will conclude And thank you for watching my 25th video. And if you are interested to to know more about plant physiology, plant embryology, plant taxonomy, mycology, or many more to come in future, kindly subscribe my channel, like it and share it. Thank you for listening.